In 2007, we presented evidence that a generic drug with a very simple structure, resembling vinegar, had the ability to decrease cancer growth by changing the metabolism of cancer cells in the laboratory. At that time, we were one of the first groups to suggest that metabolism could be the target of cancer therapies. Over the following 2 years, our work was confirmed by the laboratories of many research groups across the world, including Stanford, MIT, Harvard, University of Florida, and others. It was clear that despite the lack of support from the industry, because this is a generic drug, we had to test it in humans. After securing strong support from the university and the Alberta Health Services, we took on the challenge. At first, we collected glioblastoma tumors from 49 patients, and we studied them within minutes from their extraction in the operating room. We showed that when given DCA under the microscope, there was evidence that the cancer mitochondria function would normalize, as our 2007 work had suggested. Importantly, DCA did not have any effects on non-cancer brain tissue that we were obtaining from epilepsy surgery. This strengthened the rationale that this drug might have effects in actual patients. We then proceeded to treat 5 patients with advanced glioblastoma, in whom we secured tissues from before and after DCA therapy. This is very important. We were able to show that DCA killed cancer cells by altering the tumor metabolism, but also that DCA killed glioblastoma stem cells, the cells most resistant to therapies and the ones responsible for tumor recurrences. We were able to show that at a dose that does not have significant toxicity, there was evidence for some clinical benefit as well, although this study was not designed to show clinical efficacy or benefit. So the importance of our work it can be summarized with 3 points. Number one, it makes a small but critical step in bringing this drug closer to the patients with glioblastoma. Due to the small number of patients, of course, we cannot make any definitive, conclusive statements on whether this drug is safe or whether it's effective. However, we have acquired information, secured information to proceed to the next level of trials that will be designed specifically to show whether this drug works or not. Number 2, this is one of the first human studies in cancer that strongly points to metabolism as the target of cancer therapy. The editors of Science journal, the biggest science journal in the world, had called cancer metabolism as one of the hottest areas across all scientific fields for 2010. By studying the mechanism of the drug in human tissues before and after treatment, we can point out to several other targets within metabolism that can be used for other drug development projects. The scientists from MIT wrote an editorial about our paper, consider that a major strength of our studies. And because it's important to hear what the other people say about our work, Joan has copies of this editorial for anybody that is interested. And number 3, our work challenged the dogma that you cannot proceed with human trials unless you have industry support. We hope that this work may inspire other scientists to develop translational clinical research with other generic drugs as well.
Thank you, Evangelist. In summary, I'm going to talk about glioblastoma multiforme or astrocytoma grade 4. This is the same tumor. It's a primary tumor of the central nervous system that occurs in approximately 23,000 people annually in the United States and Canada. This is a very, very aggressive and a highly feared tumor. And so we have a long way to go with regards to developing therapies. Current standard therapy, for example, and that's surgery, radiation therapy, followed by chemotherapy, the time to tumor progression is approximately 7 months. So in other words, even on therapy, these people start to progress with their tumors at 7 months. The mean survival rate of this tumor is 14.6 months. So that's not a very good, very good figure. The 5-year survival is less than 10%. If you live more than 10 years, there's probably divine intervention. So we have a long way to go. These are very highly heterogeneous tumors. They're very unstable, and the difficulty is that as we're treating these patients, we're actually treating a moving target with mutations that are continually going on during therapy. It's very difficult to capture the tumor in a static phase, so this makes treatment very, very difficult. Now, the Cancer Genome Atlas Research Network out of NIH has recently shown that glioblastoma represents really 4 different genomic tumors. So this is why focused therapy has failed in the past, because you have to have a more holistic approach to try and solve this problem. And I think metabolic therapy, targeted metabolic therapy, focusing on mitochondrial function is one way that we can get around this so that it's not overly specific. The 5 patients that we treated in this study and that we report on, the first patient actually died at 3 months into therapy. And this was a time, excuse me, before we noticed that it takes about 2 to 3 months for the therapeutic level of DCA to appear. So these patients are quite vulnerable in the first 3 months, and we have to be very, very careful. And this is one of the good things about our scientific studies, that you can do these pharmacokinetic studies to find these factors. So it does take 2 to 3 months to get therapeutic levels. So the patient had a big, big tumor, 8-centimeter tumor, and really didn't have much of a chance. The other 4 patients survived the 18 months. One was incapacitated because the tumor occurred in the motor area and rendered very early in her disease hemiplegic. And she remains alive, but is incapacitated from that standpoint. One patient died at 24 months. This is after he had received standard therapy of surgery, radiation, and multiple chemotherapies, and then was placed on DCA and lived for 24 months, which is quite impressive. 2 patients are doing very well. 1 patient is without disease, and 1 patient is was admitted last week, and I operated to remove a tumor cyst at that time, but she's stable. So we can't make any statements with regards to the efficacy of this type of therapy, but we are enthused with what we're seeing with this very, very aggressive tumor. Thank you.
It's very important to recognize that a large number of people work together for this work. Normally, industry teams that include hundreds of people do the huge amount of paperwork required for these trials. These include regulatory bodies like Health Canada, human ethics committees, legal teams, financing or development teams, and this brings the discovery from the lab to clinical trials. We did that all ourselves. We were impressed by the public response for donations through our website, and we received checks ranging all the way from $10 to thousands of dollars from individuals or groups of people from all over the world. This work would certainly not have been made possible without the support of the University of Alberta and Alberta Health Services. We're grateful and proud to complete this small but important step for a potential treatment of a deadly disease that is supported so strongly by the people of Alberta and its public institutions in so many ways.